Welcome back. Today's lesson will be over who uses biotechnology. So, in this lesson, we will look over different industries and a brief overview of what each industry is and how they use biotechnology. So, who uses biotechnology? And biotechnology is used across many industries, and these include life science research, healthcare, agriculture, food industry, manufacturing. We also see it used in national defense, anthropology, forensics, and nanotechnology. These are the 9 different industries that we will be looking at today. So, first, we're going to look at life science research. So, life science research and biotechnology, we can see life science and biotechnology are closely connected. We have three different types of research. We have basic research, and this asks, how does life work? We have biotechnology research that asks, how can we solve a problem? And then we have applied research. So this is using science to find solutions. And an example of an applied research is virus research. We saw this with COVID. So a new virus infects humans. Basic researchers study how the virus works and its genes and proteins, and then applied researchers use this information to make drugs or vaccines to combat this virus. So there's a couple of different tools that we can use in life science research. We have cell cultures, cell expression techniques, and then we have recombinant DNA. So cell cultures are studying how cells grow and behave. Cell expression techniques see how cells make that protein, and then recombinant DNA technique is discovering what gene and proteins make or do, like what do these things do, and then we can modify those to do what we want them to do. So, what is systems biology, right? So, we can look at genes and proteins. So, we can shift from genes and proteins to the whole system. So, systems biology is studying how cells, organisms, and ecosystems work together at a molecular level. And this is made possible by advances in biotechnology. So we can look at how one cell in our body functions and how that connects to make tissues, organs, organ systems, and organisms, and how that one cell can affect our entire organism. So there are in life science research, there's a bunch of different fields that you can look at. So we have genomics, which is all DNA, so the entire genome. We have proteomics, which is studying all proteins. We have transcriptomics, which is all RNA messenger proteins. We have metatomic, which is all cell chemicals and metabolites. And then we have microbiomics, which is the study of all microbes living in a space, so the microbiome. And these studies are important because omics research makes a huge amount of data, and then that data can be processed using computers to gather and store that data, to sort and analyze it, and protect how cells and organisms will behave. And this study is called bioinformatics. And bioinformatics are bioinformaticians, so people who study this bioinformatics, are key players in modern biotechnology. We can't just make a product without knowing everything else behind it, all of the data behind the original organism and what the protein needs to look like and be going forward. So let's look at how biotech is used in healthcare. So biotechnology in healthcare is the largest part of the biotechnology industry. So products include biopharmaceuticals, medical devices, and diagnostic tests and tools. Like I said, biotechnology and healthcare are interconnected almost on a daily. So one way healthcare uses biotechnology is through drug discovery. So drug discovery uses biotechnology tools to find new medication. Companies study cells, proteins, and genes to develop possible treatments. These tools help create new tests and test new drugs. So the goal is to find a safe, effective molecule to treat a disease. You start with identifying a target and then move to a safe product. It involves both basic and applied research. So, what is the drug development process? So, we can see first we test in labs on animals, and then we test on humans in clinical trials. Then, in about 10 to 15 years, and with over $2.5 billion spent, the drug is produced and reaching patients. But most drug candidates fail before reaching these patients. They typically fail in the first stage, but we can see it fail even in clinical trials. So let's dive in and look at this process closer. So step one, we find a target and we identify it. So scientists look for a gene or a protein involved in the disease. So we can look for proteins that cause tumor growth, a gene mutation that leads to an disorder, and then we confirm that the target gene or protein really influences that disease. Once that disease has been, once that target has been identified, we can then go to high throughput screening. So this uses automatic tools to test thousands of macromolecules to see if it actually happens. So common techniques to test these are called microbe arrays and microfluidic arrays. And we'll talk about these more over in chapter 6. The goal is to find molecules that bind to or affect the target. So we take that target gene, we multiply it through PCR, and we put it in these plates, and then we wash it over with a bunch of different macromolecules to see which ones affect that gene, and then we can narrow down our tests from there. Once we find a lead compound or the most promising drug candidate, scientists ask, how tightly does it bond to the target? How does it change the target's behavior? And how does it affect the disease symptoms? So once we see that, we can then start working on producing that drug. Step 3, we then optimize. So once we find that lead component is optimized, chemical engineering is used to tweak the molecules to improve stability, absorption, or the delivery method. And it also looks at reducing harmful side effects to humans. Simulations and lab tests predict how it behaves in the body, and then we move on to trials. So we move on to preclinical trials. So that lead compound is tested in cell cultures and animal models. The compounds with toxic effects or poor performance are eliminated. And then once they have moved from the pre-trials, we go to clinical trials. Phase 1 in clinical trials asks, is it safe? We get about 20 to 100 healthy people to see how safe it is. Phase 2 is, does it work? So 100 to 300 patients are in this clinical trial to see if this medication actually works. And then phase 3, is it better than current treatments? So we have about 1,000 plus patients to see if current treatments are more effective or if this new drug is more effective than current treatments. And then, only then, after phase 3, can the drug apply for FDA or Food Drug Administration approval. So now that we've talked about drug design and discovery, let's talk about what protein-based drug production is. So, protein-based drug production is where the biopharmaceutical companies use protein-based drugs that are made from living cells. These are larger and more complex than smaller molecule drugs that might not be produced by biopharmaceutical companies. The production is expensive and complex. things that are made this way is insulin and human growth hormone. So why proteins? Why do we use a protein? Proteins like insulin are used to treat chronic conditions like type 1 diabetes, and they can only be produced in cell culture using biotechnology tools, right? So we have to make insulin. Like, we can't just give someone synthetic insulin that doesn't really work. It's not super effective. We can produce human insulin through E. coli bacteria. So it produces an active protein effectively and cost efficiently. So bacteria is most of the time used in protein production, right? I've talked so much about E. coli and it being used to make insulin. And some of the pros to this is it's fast growing and it's a simple system. It does not take a lot to grow E. coli. In fact, you could probably go swab the school toilet and put it on differential media and get a wild E. coli sample there. The cons is it's a low protein yield, right? The E. coli bacteria is very small. It's not going to produce a whole lot of insulin per one bacteria, and it cannot make high complex protein structures. And it is expensive at a large scale bioreactor system. So it's expensive to make insulin. That is why insulin is so expensive. So other expression systems. So if bacteria is not sustainable, companies can use other different systems or organisms to produce these proteins. So we can see yeast cells, plant cells, whole plant cells, animal cells, and transgenic animals all used to make these proteins. They are more capable of producing complex active proteins than E. coli might be. So they use something called a downstream process. So after the cells grow and the proteins then are extracted, purified using filtration, and then tested for activity and purity and safety. So we can see the downstream integration process here, right? Provisions of input, production, bulk cleaning and grading, processing, packaging, export, retail. So we make the E. coli with recombinant DNA or genetic engineering. It produces the insulin. We clean and extract and purify that insulin. We process and package that insulin, and then we export it or move it to the retail or to the pharmacies, and they sell it to the customer. That is the downstream processing of the protein-based production. So the final step in protein-based drug production, so once the step is purified, packaged, or purified, tested, these are packaged and shipped. Throughout the process, we have quality control that ensures FDA specifications are met so that the drug is safe for human consumption, and then safety and effectiveness are monitored at every stage to make sure that the drug will help the people in need. Biotechnology can also be used in personalized medicine, and personalized medicine is treatments tailored to the individual. This is also called pharmacological genomics, and this is where we're using the genomes or genomics to guide drug therapy. The goal is to maximize drug effectiveness and minimize the side effects to the person. So, why are we interested in personalized medicine? Well, not everyone's genetics are the same, so some drugs interact differently with these people. So individual traits affect drug response. So race and ethnicity can affect how the drug is absorbed. Gender can affect how the drug is absorbed, and so can family history. Soon, treatments will be based on individual genotypes and not just symptoms. So, genomics at the center is the basis for personalized medicine. So, a genome sequence genome sequencing is becoming affordable. So you can go get your entire genome, all of your DNA sequenced, and treatments can be matched to that full genome. We can also take tumor DNA, because the DNA in a tumor is different than your DNA, and specifically target um, cancer positive cells only. So we can see this with HECUPEN, and it targets the HER2 positive cancer only cells and doesn't affect cells that doesn't have that HER2 positive cancers. Like, if it doesn't have that cell, that target, then it's not going to affect it. We can also see gene editing for inherited diseases. So techniques like CRISPR allow precise gene editing for potentials to cure genetic diseases like hemophilia, sickle cell anemia, cystic fibrosis, and genetic blindness. We can also see personalized immunotherapies being produced where we use the immune system to treat diseases. So the CAR T cell therapy, where these T cells are collected from patients, engineered to attack a specific cancer, and then the cells go in and target those cancer cells, and it minimizes harm to healthy cells. This is FDA approved for children with acute lymphoblastic leukemia and adults with certain lymphomas. So, in the future, genetically personalized organs are looking at being made. Actually, my stepbrother produces genetically engineered corneas. That was his doctoral thesis, and I believe he's still working on that. Research into animal organs, donors like pigs, where these organs are genetically modified to remove immune stimulating markers, and human genes are added to prevent blood clots. These also can help remove harmful viruses. The aim is to reduce rejection and eliminate immunosuppressants when these organs are transplanted to humans. So we can also see biotechnology used in clinical diagnosis. Um, Clinical diagnosis use biotechnology to detect diseases and conditions in the doctor's office, hospitals, and medical laboratories. The purpose of this is to provide fast, accurate diagnoses to guide treatment. So traditional microbiology diagnosis, where we take a biological culture, and is cultured in a traditional method. These bacteria are grown to be identified, and this can take days to weeks. It is used to determine the cause of an infection for an appropriate treatment. The one major downside to this is not all human disease can be cultured in a laboratory. We can see here that, right, obviously this microorganism can be, but a large number of them cannot be. So, biotechnology has aimed to improve speeds. So, new biotechnology methods reduce diagnosis time. It can take days to weeks, and now can take hours, even minutes. We can use something called PCR-based testing to detect bacterial DNA within hours. We have specialized media that can identify bacteria quickly. And then, like I said, the goal is diagnosis in hours or even minutes instead of days to weeks. We also can see antibody-based diagnostic tools. So antibodies are proteins that bind to specific antigens or a target molecule. We can see these in dipstick tests like pregnancy, HIV, COVID tests. We can also see these in ELISA tests, which are lab-based assays that use sample types like urine, saliva, and blood to run these antibodies to see what is in that sample. We can also see quantitative lab diagnosis, so more advanced tests measure levels of targeted proteins, and this may require specialized lab instruments. So not just a PCR machine, but other equipment. Determining disease severity or stage of the we can also monitor process progression and treatment response. Advances in biotechnology diagnosis. So we're getting faster results, earlier treatment, more accurate or better targeted therapies. We can detect low levels of bacteria and proteins quickly, and some tests can even be self administered at home. So I'm going to end here and we will pick up our next lecture over agriculture. I will see you next time.